Hello again and welcome to part 2 of lecture 3 of the histology of the urinary system. In this lecture, we will discuss the histological feature of the urinary transport passages. These passages are a series of structures specialized to store and pass the urine to the exterior of the body, which means that they don't share in the process of formation of urine. They only transport the already formed urine by the kidney into the exterior of the body. These transport passages consist of many parts. We have minor calyx, major calyx, renal pelvis, which is this area here, ureter, urinary bladder, and urethra. All of these passages in general have the same histological structure, except the distal part of the urethra. In that they have, their wall is formed of 3 layers named mucosa, muscularis layer, and adventitia. These three layers can be seen in all the urinary transport passages with some regional differences. So we will start with the first layer that line the wall of the transport passages, which is called the mucosa. And as you already know, the mucosa consists of 2 layers, a surface epithelium and underlying lamina propria. The surface epithelium in the urinary system is called urothelium or called transitional epithelium. This epithelium is stratified epithelium composed of several cell layers. That's to say, it starts, for example, in the minor calyx as 2 cell layer epithelium, and this gradually increased to be 4 to 5 cell layers in the ureter, and even 6 or more layers in the urinary bladder. Essentially, this epithelium is impermeable to salt and water. So this epithelium doesn't share in any process of reabsorption of water or changing the composition of urine. These transport passages are only a conduit to transport urine from the kidney to the exterior. If we want to describe the transitional epithelium, we can say that it is organized as 3 layers. We have a single layer of small basal cells resting on a very thin basement membrane, and the second layer is an intermediate region containing from 1 to several layers of cuboidal or locolumnar cells, and we have the third layer, which is a superficial layer of large elliptical umbrella cells, which is sometimes binucleated, and these cells are highly differentiated to protect the underlying cells against the hypertonic acidic urine. Below the surface epithelium, we have a dense collagenous lamina propria throughout the excretory passages. This slide is taken from the histologyguide.com that we use for the practice of the urinary system. As you can see here, this is a section of the bladder, and we are magnifying a part of the surface epithelium. This is a transitional epithelium. And as we can see here, it is composed of 3 cell layers. The basal layer consists of cells sitting on a basement membrane, and intermediate layer consists of several cell layers, and we have the superficial layer, which is called the umbrella cells. These umbrella cells are large, polygonal, dome-shaped cells lining the lumen of the transport passages of the urinary system. The second layer forming the wall of the urinary passages is called the muscularis layer, which means that we have muscle tissue in this layer. This layer lies beneath the lamina propria, and it is composed from 2 layers. We have a longitudinal layer, which is the inner layer that is arranged in a loose spiral pattern in the ureter, and we have the circular layer, which is the outer layer, and it is arranged in a tight spiral pattern. We can see here, this is the inner longitudinal layer, and it is arranged as a loose spiral. So in histological section, in cross sections of the ureter, we can see this as if it is longitudinal. However, these turns of the muscles will be tight helix in the outer layer. So in a cross section, it appears as if it is a circular layer. And the third layer that form the wall is the adventitia or serosa. So now we start with the regional differences that we can see in the histological features of the urinary passages. The first part, as you know, is the ureter. Each ureter conducts urine from the renal pelvis to the urinary bladder, and it's approximately 24 to 34 centimeter in length. The ureter is lined, as we can see here, by transitional epithelium that we already spoke about, and in the ureter, the smooth muscles is arranged in two layers. We have inner longitudinal layer and outer circular layer. So this is a cross section showing an irregular lumen of the ureter lined by transitional epithelium. This is a lamina propria, dense connective tissue lamina propria, and inner longitudinal, outer circular, smooth muscle fibers. The adipose tissue, vessels and nerves embedded in the retroperitoneal space form the adventitia of the ureter. Next part is the bladder, which is a distensible reservoir for the urine located in the pelvis. The bladder wall, as you can see here, is thick and folded when the bladder is empty, and thin and smooth when the bladder is distended. The mucosa of the urinary bladder is formed from transitional epithelium, as we already discussed. Here it can be seen, the transitional epithelium, if we magnify the slide. This transitional epithelium, as we already said, is composed of three cell layers: the basal cell, intermediate layer, and we have the umbrella cell lining the surface of the bladder. These umbrella cells are especially well developed in the bladder because the contact of the bladder cells with the urine is greatest in this area, and the urine stay for longer period of time than in the ureter or urethra, for example. And that's why we need a very specialized cells that will protect the epithelium, the underlying epithelium, from the acidic nature of the urine, and umbrella cells will serve this function very well. We have a highly vascularized lamina propria underneath the surface epithelium, and this is followed by a very thick muscle layer. As you can see here, the bladder has a very thick muscular wall. The muscle of the bladder is called the trousseau muscle, and it's composed of inner longitudinal, middle circular, and outer longitudinal muscle. However, the delineation of these layers are not very clear, and as we can see, they all function as a single unit, and when they contract, they forcefully expose the urine out of the bladder. The last part of the transport passages of the urinary system is the urethra. The urethra is a fibromuscular tube that conveys urine from the urinary bladder to the exterior through the external urethral orifice. The size, structure, and the function of the urethra differ between males and females. However, the urethra is lined with the same epithelium in the male and female, and as you can see here, the lumen of the urethra is very distinctive because it has these longitudinal folds which can give the urethra a very characteristic appearance. So we will start with the male urethra, which serves as a terminal duct for both the urinary and genital system. Male urethra is about 20 centimeter long and has three distinct segments: prostatic urethra, membranous urethra, and penile or called spongy urethra. The prostatic urethra is the first part which extends for 3 to 4 centimeters from the neck of the bladder through the prostatic gland, and it is lined by the transitional epithelium, the same mucosa that line the bladder. The second part is the membranous urethra, which extends for about 1 centimeter from the apex of the prostate to the bulb of the penis. This part of the urethra is lined by stratified or pseudostratified columnar epithelium, which is similar to the epithelium lining the genital duct that you will discuss it further when you take the male genital system. The final part is the penile urethra, which extend for about 15 centimeters along the length of the penis. This part is lined with pseudostratified columnar epithelium proximally. However, at the distal end of the penis, near the exterior, it is lined with the stratified squamous epithelium, which is continuous with that lining the skin of the penis. The female urethra, however, is shorter, much shorter than the male urethra, and it measures only 3 to 5 centimeters in length, and it is initially lined by transitional epithelium, and this epithelium gradually changes to non-keratinized stratified squamous epithelium continuous with that of the skin at the labia minora. And the female urethra, you will discuss it further also at the female genital tract. So, as a summary of our 3 lectures, we can say that the urinary system is one of the most important systems in our body. It receives about a quarter of the blood ejected from the heart. The main function of the urinary system is to maintain the homeostasis of our extracellular fluid and the plasma, of course, remove waste products, and maintain acid-base balance. This occurs through absorption and secretion of different substances in the renal parenchyma during the process of plasma filtration. The initial filtrate will start in the glomerulus, where it passes from the urinary space into the proximal convoluted tubule, and then pass into different channels or tubules, where each one of them has a specific function to concentrate or to change the composition of this filtrate. It is very evident from this photo that each segment of the nephron has a very specific function. Some with the absorption of sodium, for example, or potassium, or chloride, or reabsorption of water, removal of waste products, for example, urea, removal of urea from the renal interstitium, reabsorption of amino acid. All these processes occur, whether can be active or passive processes, occur continuously in the renal system to maintain the health of our body. Thanks for listening, and I hope that you enjoyed our lectures, and see you in the next system.